Hi, I'm Rich with insideHPC. We're here at the DDN booth at SC12 in Salt Lake City. I'm here with Skip from the Virginia Bioinformatics Institute.
Absolutely.
Did I get that right?
Yes.
Okay, so Skip, let's just start at the beginning. Who is the Institute and who do you help?
Well, the Virginia Bioinformatics Institute is at Virginia Tech. It's about 320 people currently and growing. And we do bioinformatics in all different flavors. We have divisions that do large-scale models and simulations and things like infectious disease, and we provide input directly to the White House, for example, during large things like the swine flu. You know, do you close airports? Do you close borders? Things like that. And then we have other institutes that do everything from plant science to human genome research, which is my particular endeavor. I spend my time doing researching and comparing normal genomes to cancer genomes, and it allows us to make new discoveries that we can translate directly into the clinic.
Well, this sounds like really data-intensive kind of work. Is that the case? Does it take a lot of I/O to do what you guys do?
Oh, absolutely. So there's a couple of things that's happened very recently that's really kind of changed the way in which we can do some science. The first thing that's happened is there's this new generation of DNA sequencers that allows us to get DNA out, sequences out for human genomes at $1,000 instead of $3 billion. So what's happened is now there's literally thousands of genomes that are available. The other thing that's happened is enough supercomputing power to actually analyze them all at once. And the third thing was we came up with some cool algorithms that allowed us to do it accurately and precisely. So what do we do now? We've actually looked at thousands of human genomes that are people without disease, and we compare it to people with disease, and we actually compare it in unique areas. I look at junk DNA, okay, where lots of people don't tread. And so we actually make our discoveries in places where people haven't looked before. So we look at places where, you know, the normal human would have, say, one set of DNA sequences, and cancer people would have something that's entirely different. When we discover something like that, it says, aha, we've got a chance to maybe find something that's a new diagnostic or a new way to determine whether a cancer is aggressive or whether a therapy will work or a new drug target. We do all those things.
Okay, so the way it sounds, you're comparing these must be big files that you're working with.
Well, every human genome is about 1 terabyte. Okay, and so I think, I lose track of this, but I think we've got somewhere between 4,000 and 5,000 right now. So you can do the math. And so at any given time, I would say we probably have 400 or 500 terabytes online that we're working with, and then rolling those off into, to storage. But it really means we've got to have those going and hot and active. And so only having lots of storage, lots of computing power, lots of bandwidth allows us to do this.
So how do you architect the storage piece? I'm really interested in how can you feed a beast like that?
Well, we actually had to build a machine. It's called Shadowfax, actually, and it's a heterogeneous computing environment. So it's got regular processors and graphics processors and gate array processors. But then the big thing about genome science is that it's very data intensive. So we have to worry about bandwidth to that, and then we have to worry about lots of storage. So I'd say we have somewhere, just the storage of kind of working files close to a petabyte on spinning disk, and we have a 50 petabyte storage system for tape. And we're keeping up. And the other thing is I think we use about 25% of the total bandwidth that comes into the university just to keep up with downloading all the genomes that are being produced everywhere else. And so once it's all there, then it's just up to my nerds, my hackers, my programmers to boil it all down to, for me, a spreadsheet that says, here's the places in the genome, here's normal, and here's cancer. And so then we go back into the lab, verify that it works, taking real DNA samples, testing it, and if it works, boom, we try to get it out into the clinic. So get it out of the university, into a company, actually the company's Genomion, and then its role is to go out and negotiate with these clinical testing companies and pharmaceutical companies to take these advances and get them to the clinic as soon as possible, because every day you save you save a lot of cancer patients.
Well, that's terrific. That's
Yeah.
exciting. So I've always been curious, Skip, about the signal to noise ratio when you got the big files like this in genome. Are you looking for the virtual needle in the haystack, or is that the wrong analogy?
Well, it's really weird that you bring this up. So actually the big thing that enabled us to do this was that all the genome assemblies at about 1 million places. So the genome's got 3 billion letters. There's about 1 million places in the genome where the sequence is not simple. It's repetitive, and it's sequences like CAG, 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 CAG. Well, our cells make mistakes and they stutter when there's things like that. And so because they're repetitive like that, they're extremely hard to accurately get correct with the current algorithms. So that was actually our secret sauce, and that was we built algorithms that raised the quality from 20%, the accuracy from 20% to over 95%. So if you have 80% noise, you can't find a correlation. And so when we got to 95%, then we started, aha, we've got it. Now we just have to calculate, 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 and overlay the data, and it just comes out.
That's great.
It's critical, in other words.
Well, Skip, hey, thanks for sharing that. This is really
Absolutely.
exciting. Congratulations on your work.
Yeah, well, I just certainly hope that that we can get it from the computer to the clinic as fast as possible, because these are it's critical for people to know their risk for cancer. If they know it, they can take actions to prevent it. Their doctors can adjust therapies and all the kinds of things. This is just what we do.
Awesome. Personalized medicine. Sounds like it's coming real soon.
Oh, it is. It is. And my genome's been done. We can do yours too.
Well, that's scary. Mine's probably going to get filed next to Ozzy Osbourne, but that's another story. Hey, Skip, thanks again, man. This has been great.
Absolutely. Well, thanks for talking to me.
You bet.